I'd like to welcome you all today. This is really very exciting for us. We have Dr. Sean Cowper, who is chairman of our Medical Advisory Council, and we have had pre-submitted questions on all kinds of fibrotic diseases sent in to us via our website. We've asked people to questions that have been on their minds and maybe they haven't gotten an answer to. We've given Sean all of these questions and he's done his research and he's ready now to talk about this. This is not an open question and answer period. These are pre-submitted questions. So Sean, we're excited to hear about this.
Thank you. Thank you, Gloria. Thank you to the people who are actually here in the room today. For those of you in attendance today, I'm reading from a prepared script and this transcript will go on the web. Thank you for coming. For those of you viewing this online or on the video, this presentation is being recorded at the 6th Annual Yale Fibrosis Symposium taking place in Orange, Connecticut on November 17, 2012. For those of you who do not know me, my name is Sean Cowper. I'm a physician at the Yale University School of Medicine in the field of skin pathology. I'm a co-founder of the Global Fibrosis Foundation, and I serve on its board of directors. I'm also the Medical Advisory Council chairman. I'm joined today by Dr. Ali Abu Alfa, to my left, the director of the nephrology section at the American University in Beirut, Lebanon. Dr. Abu Alfa is also a GFF board member and a member of our Medical Advisory Council. The MAC, Medical Advisory Council, consists of dozens of physicians and scientists who assist with scientific questions put to them by the board of directors. Members of the MAC assisted with the preparation of the informational session today. The Global Fibrosis Foundation was founded in 2009 as a non-for-profit organization with the goals of educating physicians, patients, families, and policymakers about the process of fibrosis and to facilitate the discovery and dissemination of new treatments for fibrosis. Last night, the GFF annual research dinner took place here in New Haven, Connecticut, and we are happy to report that a $25,000 grant was awarded by the GFF to researchers at Case Western University in Cleveland, Ohio, for a project addressing the basic science of skin fibrosis. The purpose of this informational session is to address questions about fibrosis that have been submitted by visitors to the Global Fibrosis Foundation website. We received many questions, some of which are variations on a theme. It became clear upon compiling and reviewing these questions that there are significant misunderstandings about the process of fibrosis and about the disorders characterized by tissue fibrosis. I will begin this session by first providing the answers to 3 questions that I made up to address this. These will provide a basis for understanding many of the subsequent responses. This will allow persons without a medical or scientific background to better understand the goals of the scientists and physicians working to tackle these issues. So my first 3 questions are these. Question 1, simply, what is fibrosis? When I was just about to enter college, I had the bad luck to develop appendicitis, and lucky for me, surgeons removed my infected appendix through an incision about 3 inches long and sewed me up with suture. Today that scar is flat and thin and barely visible. Many of us have had similar experiences and carry similar scars. When tissue of any type is injured intentionally via a surgeon or unintentionally as a result of a mishap with a knife or other sharp object, it must knit together again. Sometimes we require stitches to allow this to happen. Sometimes the wound is small enough that a bandage will do. At the level of the cell, the process we call healing is immensely complex and not completely understood. Successful healing requires the coordination of many cell types coming together to create new and undamaged tissue, to fight infection and to reestablish connections of blood vessels and nerves. The most critical cell for creating this new supporting tissue is the fibroblast. Fibroblasts produce 3 substances that serve as the building blocks for the supporting tissue: collagen, mucin, and elastin. You've probably heard of collagen, which is a material often added to various cosmetic products. Collagen is the most common protein in mammals, makes up 1/4 to 1/3 of all proteins in our bodies. Collagen commonly forms fibers, and that's a useful point to remember. Collagen is a fiber, and collagen is produced by a fibroblast. The image there reveals what I'm discussing. Here is an analogy for you. In the connective tissue of the body, collagen acts like bricks or 2x4s. Mucin acts something like mortar or glue, and elastin acts something like rubber bands. These three items, when laid down in an orderly way and in correct proportions, produce a framework of tissue that can be solid or soft and flexible where it needs to be. If, for whatever reason, a fibroblast produces too much of this critical building material, or if the orderly structure or proportions of the materials are not appropriate for the body site, the function of the affected tissue may be, the function of the tissue may be affected. The interference of tissue function by too much collagen is called fibrosis, and some people use the word scarring to mean the same thing. Examples of fibrosis include permanent scarring of the lung after certain types of pneumonia, scarring of the liver in cirrhosis, scarring of the heart following a heart attack, scarring of the lung in cystic fibrosis. All of these processes represent tissue injury, although the original cause of the injury can be very different from organ to organ. It is this end process called fibrosis that is common to all of these diseases. If fibrosis can be prevented or treated, the impact on the death and suffering of human beings would be tremendously reduced. The mission of the Global Fibrosis Foundation is to help those with fibrosis by understanding the process and developing effective treatment and prevention strategies to eliminate its permanent effects. Question 2. What is nephrogenic systemic fibrosis? NSF. NSF is a subject near to my heart and served as the spark that brought the Global Fibrosis Foundation into being. As the final word in its name suggests, NSF is characterized by the deposition of too many of those building blocks, collagen, mucin, and elastin, in the tissues. The chief tissue affected is the skin, although other organs may also be involved. Because this disease can potentially affect tissues in various parts of the body, it is called a systemic fibrosis. It can affect the entire person, not just their skin. My colleagues and I were the first investigators to recognize that NSF existed at all. In 2000, we wrote a letter to the editor of the British medical journal, The Lancet, that we were investigating a new disorder in which collagen was being deposited in the skin and arms and legs of people with kidney disease. The disease only affected a small number of kidney patients, but quite clearly all of the affected patients had kidney disease of some type. The word nephrogenic addressed the observation that kidney disease was a necessary precursor to the development of NSF. Why this was so was not understood in 2000. Over several years, our group and a few others continued to investigate these patients. The close examination of dozens of patients and their complex medical records by our team and teams we called in from the Centers for Disease Control led to a regular repeating pattern. All patients had kidney disease, but the underlying cause of the kidney disease seemed irrelevant. Upon examining the patients, they generally had skin thickening or hardening on the arms and or legs. In many cases, this hardening made flexion of the joints impossible. The disease often occurred rapidly and sometimes improved if the patient's kidney disease improved. Many of these patients had been evaluated by X-ray and MRI procedures. I am a skin pathologist, so besides evaluating the patient, I also evaluate their skin by biopsy. As I noted, the skin was hard and the function of the tissue was affected by this hardness. This satisfied the clinical definition of fibrosis that I mentioned a few moments ago. The story was similar under the microscope. There we observed increased amounts of collagen, mucin, and elastin, as well as numerous cells producing these materials. Why fibroblasts would behave in this unusual way without any visible injury was a mystery. The process under the microscope resembled a wound healing reaction gone mad, but there was no wound. Somehow the fibroblasts were receiving the signal to make collagen despite there being no wound. Many of you already know the rest of this story. Careful review of many patient records, some lucky observations by patients and physicians, and good scientific investigation methods eventually identified that most of the patients who developed NSF had been given a medication called a contrast agent shortly before developing the disease. The contrast agent is a chemical that allows radiologists to improve the quality of images produced in certain MRI examinations of the body. The contrast agents are produced by several companies in the US and Europe. An investigation of many such cases in many countries led the investigators to conclude that one component of the contrast agent, namely an atom called gadolinium, here, pictured on the atomic table as the red square near the bottom, was the trigger for NSF. The gadolinium was in different chemical forms in different contrast products. The large-scale investigation is still ongoing, but the evidence strongly favors that only a few such products are at a high risk of producing NSF. The U.S. FDA and the European medicine agencies made new rules regarding the use of these higher-risk contrast agents a few years ago, and since then, new cases of NSF have essentially vanished. In fact, this graphic shows you the flattening out of new cases as of last year in 2011. I can say that we've expanded this out to 2012. I continue to examine new cases as they come in, but the exposures have all been before the 2010 date on that chart. So, since then, new cases have essentially vanished. In the space of about a decade, a new disease was recognized, investigated, and effectively prevented before gaining a strong foothold in the population. In fact, many doctors have still never heard of NSF, and virtually no members of the general public have. Unless you have personal experience with this disease, or are a fibrosis researcher, a maker of contrast agents, or an attorney making lawsuits on behalf of patients with this disorder, you likely have never heard of NSF. Question 3, the last one that I authored. So if NSF is disappearing, why is it important at all? Well, number one, NSF, as I mentioned, is fibrosis without a wound. Using animal models and gadolinium contrast agents, scientists can now recreate the process of fibrosis in order to study it. There are relatively few good models for researchers to study fibrosis. The discovery of an important new model enables researchers to explore many new theories about how fibrosis starts and how it may be controlled. Number 2, NSF is a man-made disease. Medicine manufacturers and medicine approval agencies like the U.S. FDA have learned from this experience and are already adopting techniques and developing new products that address the problems that allowed NSF to emerge in the first place. Similarly, physicians and hospitals have used this experience to better control the use of beneficial agents that could be detrimental to small at-risk populations. Number 3, the patients who carry a diagnosis of NSF are affected in a variety of ways. Some are profoundly disabled, while others suffer chronic aches and pains tied to their gadolinium exposure. These patients need effective therapies. At present, no universally accepted or fully effective therapy exists for NSF. Number 4, the pharmaceutical industry is now recognizing the potential for new agents that address fibrosis. As mentioned earlier, many diseases and injuries terminate in fibrosis. Stated differently, fibrosis is the final shared pathway for many types of tissue injury. It is the end of the line, and once there are no universally effective ways to go back. The ultimate therapy will be one that stops fibrosis before it ever begins. It is entirely possible and probable that therapies that address the signals that lead to end-stage fibrosis will treat all types of fibrosis. The pharmaceutical industry will not invest millions or billions of dollars to treat a rare disorder like NSF. But if their investment will treat the fibrosis of a heart attack, cystic fibrosis, cirrhosis, idiopathic pulmonary fibrosis, it will impact the health of everyone, even those with rare fibrotic disorders. So that ends my introductory set of questions and hopefully sets the stage for discussing questions received from the GFF webpage visitors. I think the best way to do this is to read the question verbatim as received by us and to address the question and any assumptions the questioners make. I'm not sure I am supposed to reveal the identity of the questioner, so I will err on the side of caution and not do so. I should have ample time to cover these questions. Question 4, submitted to the GFF website. In quotes, we know that the lollipop lesion is identified and present in 5% of the most advanced cases in the gadolinium fibrosis spectrum. What research or tests can be done to identify the early stages of the disease before lollipop lesions appear so the patient can get early care for a better outcome? End quote. First, to address the assumptions in the terminology. It's vitally important to remain as precise as we can about what we mean and not introduce new phrases that are inconsistently defined or not defined at all. So the question refers to the lollipop lesion. This fanciful reference is to a particular finding on skin biopsies from some patients with NSF. I'll admit I coined that one myself. The finding has other less memorable names in the medical literature, such as osseous bodies, sclerotic bodies, osseous metaplasia, etc. But I can remember lollipops better, hence the name. As I mentioned earlier, fibroblasts produce building blocks of tissue that include collagen, mucin, and elastin. The lollipop I described is a blob of rounded collagen fibers with protruding elastic fibers. The appearance looks like a lollipop to me. In a comprehensive survey of all of the cases of NSF contained in the files of the registry at Yale, my coworkers and I identified lollipop bodies in 5% of NSF cases. The finding is considered uncommon, but when it is present, it is strongly suggestive of a diagnosis of NSF. There's no critical study that has showed that lollipop bodies are more common in earlier or later cases of NSF. Why they occur in some patients and not others is also not clear. The assumption that lollipop bodies indicate an advanced case of NSF is not supportable at this time. A second phrase mentioned by the questioner is, quote, gadolinium fibrosis spectrum, end quote. I have not seen this terminology in the medical literature, and I'm not sure exactly what's meant. The only disorder I am aware of that associates fibrosis with gadolinium exposure is NSF. And since that is well defined, that terminology is what I will use. The questioner does make an important point in their question. The earlier a diagnosis of NSF can be made, the better. Some therapies, such as intense physical therapy, may help to preserve mobility if introduced at an early stage of the disease. Similarly, kidney transplantation or treatment of an underlying kidney disease may reverse early cases of NSF before significant collagen is deposited. Ultimately, however, the core question is what research or tests can be done to identify the early stages of NSF. At present, NSF is diagnosed by a thorough examination of the patient, including a history, family history, review of systems, physical examination, appropriate blood tests, and biopsy of thickened skin. The procedures for this are outlined in an article that will be referenced at the end of this transcript. There is no blood test that can diagnose NSF at this time. The blood tests I refer to are to help exclude other diseases that resemble NSF. Scleroderma would be a good example of this. Following the guidelines in that article, the diagnosis of NSF is made by obtaining a compatible biopsy in a person with a compatible clinical finding and ruling out other diagnoses that may otherwise mimic NSF. Question 5. Quote, has any study been done to determine whether repeat biopsies might be exacerbating the symptoms of so-called NSF through an immune response reaction triggering release of stored gadolinium, end quote. Some trace elements, such as cobalt, nickel, and copper, have physiological roles in humans, and there have evolved methods to transport and handle these substances in the body. Gadolinium is not known to have a physiological role in humans, and consequently there is no specific transport or storage mechanism. Like many external contaminants, the body may isolate foreign materials to remove their possibly dangerous effects. For instance, many tattoo pigments intentionally introduced into humans by other humans contain heavy metal contaminants such as cadmium and mercury. Skin biopsies collected near tattoos will show cells called macrophages containing the colored pigments. This is one method the body uses to isolate and remove foreign substances. The biopsies of patients with NSF frequently contain not only fibroblasts, but macrophages as well. These have been shown to contain gadolinium in several studies, and this observation is one of the clues supporting the contention that gadolinium triggers NSF in susceptible persons. In addition, researchers have long noted that the size of gadolinium atoms is close enough to calcium atoms that gadolinium may potentially interfere with calcium function in the body. Beyond bone formation, calcium is involved with many critical functions in the human, including blood clotting, muscle function, and blood pressure maintenance. I personally believe that the influx of large numbers of gadolinium atoms may well explain other observations that have been made in patients with NSF, including unusual muscle contractions, pain, unusual nerve function and sensations, and a tendency to clot blood too readily and to have blood pressure instability. The questioner inquires whether an immune reaction might release stored gadolinium. As mentioned earlier, there is no evolved method for storing gadolinium that we know of. There is, however, a way of storing calcium, and that is in the bone. For this reason, researchers are very interested in knowing how much gadolinium is stored in the bone, how it could be released, and under what circumstances it may be released. It is reasonably safe to assume that the release would not be triggered by an immunologic reaction. It is possible, however, that hormones that regulate calcium levels may also release stored gadolinium in the bone. This is an active area of research. Question 6, quote, a European Medicine Agency safety document comments on gadolinium in bone stores, speculates that a person with normal kidney function who has been exposed to gadolinium could potentially get NSF if at any time in the future their kidneys fail. In view that gadolinium has been found in the hip bones of a person with normal kidney function 8 years after gadolinium exposure, what is the opinion of experts in the U.S. as to whether the circumstances of bone-stored gadolinium might lead to a delayed onset of fibrosis in persons with normal kidney function, end quote. As mentioned earlier, there is evidence to suggest that gadolinium may be stored in the bones, and researchers are working to understand whether a massive release of gadolinium from a bone could result in the development of NSF. Here is my opinion on that subject. We know from experience that patients without kidney disease are at no risk for the development of NSF. This is because gadolinium-containing contrast is removed from the body by healthy kidneys very rapidly. If patients with normal kidney function were at risk, there would be millions upon millions of cases of NSF, and there are not. The persons with NSF in our almost 400-person registry at Yale have kidney disease, either chronically or transiently at the time of the gadolinium contrast dose. These persons were not able to excrete their dose appropriately due to the kidney dysfunction, and as a result, contrast was able to accumulate in their tissues, where much of it was engulfed by the macrophages. These macrophages have digestive substances capable of breaking down certain gadolinium contrast molecules. Once the gadolinium is separated from the remainder of the contrast molecule, it is available to bond to tissues, get trapped in bones, and cause other problems. The European speculation referenced in the question assumes that a dose of gadolinium-containing contrast would remain in the body long enough for it to break down and be incorporated into the bones. There's no reason why that would happen if the patient's kidney function were normal. As the questioner notes, gadolinium may be found in the bones of patients with normal kidney function. I will also point out that gadolinium can be found in the skin and other tissues of patients with normal kidney function. These detection methods are extremely sensitive, and the body's excretion mechanism is less than 100% perfect. So do I think a small amount of gadolinium released from bone would likely cause NSF in a patient with normal kidney function? No. A small amount of gadolinium released from bone would be no different than a small amount administered by vein. It too will be excreted in the urine. Question 7. Quote, what is the opinion of experts with regard to the risk of non-contrast MRI causing displacement of previously stored gadolinium from the bone and triggering a fibrotic process in a person with normal kidney function, end quote. My opinion is that the small amount of gadolinium released from bone would never approach the rather massive dose that is given for a contrast MRI study. If the patient's kidney function is good, it will be excreted. As for non-contrast MRI displacing gadolinium, there's no reason to believe that this is even possible. Question 8, quote, might ordinary MRIs exacerbate symptoms in diagnosed or undiagnosed NSF, GASF patients by stirring up stored gadolinium? The administration of gadolinium-containing contrast agents is contraindicated in patients with a diagnosis of NSF. I've reviewed cases of patients who likely had NSF and didn't realize it and were administered repeated doses of gadolinium-containing contrast agents. These patients always had kidney disease. In those cases, the symptoms often got worse. As I mentioned in the prior response, there's no reason to believe that pre-existing gadolinium in the body is stirred up by MRI. Also, the questioner refers to NSF as NSF-GASF. I am familiar with the GASF terminology, but find it unnecessary, confusing, and redundant. For those not familiar with GASF, it refers to, quote, gadolinium-associated systemic fibrosis. It has been used in a variety of ways that have confused the meaning of the term. Let me explain the two ways in which it has been used and why it's unnecessary and sometimes flat wrong. One, some persons wish to use GASF to describe a patient with kidney disease who has developed skin fibrosis after exposure to gadolinium-containing contrast agents. In such cases, NSF is the accepted term. Physicians, insurers, researchers, attorneys, pharmaceutical corporations all understand that the association of NSF with gadolinium is solid and have acted on this information. Changing the name unnecessarily creates confusion, as all of these parties begin to wonder if they are talking about the same disease when the other term is inconsistently applied. One simple illustration: if insurers pay for physical therapy for NSF based on the established history of reports favoring this treatment, what will happen when they are confronted with the terminology GASF on a request for reimbursement? Number two, another use of the term GASF. Other people wish to use that to describe a patient without kidney disease who has developed unusual aches and pains after exposure to gadolinium-containing contrast. These patients do not have skin lesions or fibrosis. In such cases, and I know many of them exist, any terminology containing the word fibrosis is completely misleading, as the patient does not have fibrosis. I would suggest that these patients be diagnosed as having a gadolinium-associated pain syndrome. More about that later. Question 9. Quote, are any studies planned to evaluate the risk of any degree of gadolinium-associated systemic fibrosis in persons with normal kidney function? End quote. Here is an example of how GASF as a terminology is confusing. I cannot determine what's being asked. I will assume the questioner is asking whether there are planned studies to evaluate whether NSF can occur in the setting of normal renal function. The answer is no, to the best of my knowledge. However, many interested parties, the FDA, pharmaceutical companies, NSF litigators, researchers, etc., would be very interested if more than a few such cases come to light. Based upon my past and current experience with hundreds of cases of NSF, I personally have not seen such a case and believe I would have by now. After the identification of gadolinium as a trigger, and especially given the promise of monetary awards for lawsuits able to prove this point, one would expect such cases would be widely known if they existed. Question 10. Many people have documented toxic levels of gadolinium in their urine per the Mayo Clinic reference range, years after their last contrast MRI. Many also have medical conditions that are also seen in the secondary symptoms of NSF. What studies have been done to understand retention of gadolinium in non-renally impaired patients, especially with regard to patients who have multiple MRIs? And what are acceptable levels of gadolinium that will not create a chronic condition similar to but different from the acute condition called NSF? End quote. I personally am not aware of publications or studies addressing high levels of urinary gadolinium years following exposure. I am aware that this topic is frequently discussed in GASF chat rooms, however. The finding of high gadolinium levels without a known source of exposure is troubling and should be investigated. In light of the relatively brief half-life of gadolinium-containing contrast agents in persons with normal kidney function, any measurable gadolinium level in the urine is worth investigating. Whenever unusual trace materials are present in the urine or blood without explanation, it is worth, sorry while I change my page here, it is worth conferring with an epidemiologist or toxicologist to identify possible sources of contamination. Potential sources could include environmental or work exposures via inhalation or ingestional routes or contamination of food. The gadolinium contrast manufacturers are responsible for conducting studies regarding the excretion and metabolism of their agents. The FDA requires this for approval of the agents. I do not believe these studies have been repeated in recent years, although one could make a good argument for doing so. I am not aware of anyone having determined a safe or unsafe level of gadolinium, since no level of gadolinium would be considered normal in an unexposed individual. A safe range of values would be difficult to define. Of course, in a patient who has had MRI contrast, high levels of urinary gadolinium would be normal, as this is how the material is shed from the body. This would be expected to taper down to an undetectable level over a period of time that is dependent on the kinetics of the drug. Question 11, quote, much has been written about the skin manifestations of NSF, but less information is available concerning the systemic fibrosis that continues to get worse as the disease progresses. What research has been done about the systemic progression of the disease that can be tested for on patients who have documented toxic levels of gadolinium but have not developed the skin symptoms? End quote. For patients with skin NSF with internal involvement, there have been little information regarding the internal progression because few patients have been followed prospectively, and documentation of internal fibrosis is very difficult. In such cases, worsening of the skin would be an external indicator of what is likely going on internally. The questioner also asks about patients with, quote, toxic levels of gadolinium, end quote, who have systemic progression but do not have skin changes. At present, the diagnosis of NSF requires a compatible clinical picture as well as a supportive skin biopsy. If a patient has no skin symptoms, they cannot have NSF as it is currently defined. In addition, the presence or absence of gadolinium in the tissues or the urine does not impact on whether the patient does or does not have a diagnosis of NSF using that definition. If a patient has internal fibrosis that is affecting the function of organs but does not have skin changes, they would need to have a full medical examination to document the extent of injury, including blood studies to exclude rheumatologic disorders like scleroderma and mixed connective tissue disease. Question 12, quote, we understand that there are specialists for specific types of cancer, heart disease, etc. How can a patient with GASF-NSF get referred to an experienced MD for treatment? End quote. It is challenging to find experienced NSF physicians and still more challenging to get effective treatment. I would strongly suggest you contact the International NSF Registry at Yale for guidance. We maintain a list of physicians who have familiarity with NSF and can direct you to the closest centers for treatment. Unfortunately, some of the most experienced physicians with NSF have only encountered a handful of patients with the disorder. It is very rare indeed. Question 13, quote, what new research is being done to find new treatments, end quote. There is no coordinating body for NSF research. We do not necessarily know what other centers have planned or are pursuing unless they contact us in advance. Additionally, we do not know what studies are underway until they are presented at conferences or published in medical journals, at which point the study has usually closed. As for research conducted within the pharmaceutical sector, there is minimal interest on their part to specifically target treatments for a disorder that is fully preventable and very rare. However, there is great interest in learning about the process of fibrosis as a final common pathway in numerous common conditions. It would therefore appear that the best hope for an effective NSF therapy is to support research studying the process of fibrosis, then piggyback onto their success. Each breakthrough for the treatment of scleroderma and idiopathic pulmonary fibrosis represents a potential therapy for patients with NSF. Question 14. Quote, gadolinium-based contrast agents have been found to stimulate pro-fibrotic and pro-inflammatory responses. Have any retrospective studies been done to determine if patients diagnosed with a fibrosing disorder had prior contrast-enhanced MRIs? End quote. I agree with the first statement that gadolinium-based contrast have been found to stimulate a pro-fibrotic response. That's just another way of saying that gadolinium-containing contrast agents may trigger NSF in susceptible persons. The Yale NSF Registry has investigated this question and found that a gadolinium exposure can be found in most cases of NSF, and that in the best documented cases for which we have detailed records, an exposure to gadolinium-containing contrast agents occurred within 5 weeks of the onset of NSF in half of the studied cases. The contrast agents involved tended to be those with a linear chemical structure, precisely the ones that have since been contraindicated in kidney patients in Europe and the United States. Question 15. Quote, studies indicate that gadolinium is nephrotoxic and can cause acute kidney injury. As AKI can happen to anybody regardless of their normal kidney status, what considerations have experts given to any possible risk of gadolinium-induced transient AKI leading to a fibrotic process akin to NSF in a person with usually normal kidney function, end quote. There have been patients with AKI who received gadolinium-containing contrast during this acute and transient phase and subsequently developed early changes of NSF. Every such case I am aware of improved spontaneously or disappeared completely once the transient kidney injury resolved. My experience has been that if kidney function can be fully reestablished within approximately 1 year of NSF onset, the patient's NSF will spontaneously improve and may fully clear. It may take months for this to occur, however. Question 16. If a patient has extreme burning or pain shortly after an MRA, that's a magnetic resonance angiogram, what protocol would be recommended to radiologists or emergency room doctors as a remedy to help the patient's suffering and prevent oxidation from toxic gadolinium? End quote. Patients who receive these agents may have transient sensory symptoms such as burning and pain. A small number of these patients seem to develop a more severe and prolonged pain syndrome that may require narcotic medication for relief. I refer to these patients as having gadolinium-associated pain syndrome. This is not a term in the literature. This is how I refer to them currently, but it will be in the literature in time. They do not fulfill the criteria for a diagnosis of NSF as they have no skin changes and no fibrosis. Compared to the large numbers of contrast MRIs performed, these patients seem to be very rare, and the reasons why they have these symptoms are at present unknown. Interestingly, many patients with full-blown NSF also have a similar pain syndrome, although many with NSF describe no symptoms at all. It may be that NSF and GAPS are two independent processes that are both capable of being triggered by gadolinium-containing contrast, and some patients are unfortunate enough to be afflicted by both conditions. The nature of GAPS is not understood. To our knowledge, there is no measurable finding that can be used to verify the sensations the patients purport to have. I expect that intensive study by chronic pain or neurology specialists will be necessary to fully evaluate this condition. The questioner inquired what protocol should be followed to help these suffering patients. There is no protocol. At present, patients suffering with this type of pain are treated by pain specialists using a variety of agents, typically used in the treatment of chronic pain syndromes. The therapy will likely be highly individualized. Question 17. Quote, researchers have estimated that at least 1% of gadolinium from each dose of a gadolinium-based contrast agent remains in the patient's body, even in patients with normal renal function. It's also known that GBCAs and free gadolinium both stimulate normal fibroblasts. Since gadolinium is known to deposit in bone and remain in the body for long periods of time, what are the long-term risks from the accumulating 1% from multiple doses? And have any studies been done to determine how much gadolinium it takes to initiate the internal fibrosis associated with NSF, GASF? End quote. Detailed pharmacokinetic studies regarding the amount of gadolinium deposited in bone and how it is released are not available in the public literature to my knowledge. As the questioner mentions, there have been studies on cultured fibroblasts that indicate increased collagen production upon exposure to gadolinium-containing contrast agents. These have been conducted on cultured cells under controlled conditions. The findings may or may not be relevant to what is actually happening in the complex environment of the human body. More complex modeling with mice and rats have also addressed some of the fundamental questions about gadolinium within a living organism. Such studies typically test a range of doses that are meant to simulate gadolinium contrast doses in a human. testing within a living human under circumstances that are contraindicated by the FDA would be considered unethical. When I presented data from the International NSF Registry to the FDA, I pointed out that many patients developed NSF with a single dose of gadolinium contrast. I also pointed out that many NSF patients were dosed many times before they actually developed NSF. These observations suggest that in susceptible persons, nearly any dose could be problematic. The difficulty is determining what unique factors in a given individual will trigger the disease. Question 18, quote, research indicates there may be cofactors to gadolinium in the development of NSF such as acidosis, inflammatory factors, and transmethylation. All of these can happen to anybody. What consideration have been given with regard to any risk to a person with normal kidney function developing NSF, or more appropriately named gadolinium-associated systemic fibrosis, through the existence of one or multiple comorbidities in the setting of gadolinium, end quote. As I mentioned previously, there have been many observations over the years regarding comorbidities or concurrent conditions that a patient may have that could promote the development of NSF. Some of these were speculation, some were likely coincidental, others turned out to be core features of the disease. When Dr. Philip Lavoie and I first investigated NSF, before we even had a name for it, one thing was clear. It was happening in patients who had had a kidney transplant. This initial impression turned out to be false, however. More searching turned up patients who were being dialyzed but had not been transplanted. So we revised the hypothesis, speculating instead that the common denominator might be dialysis, until we started finding people who had kidney disease but had never been dialyzed. And at that point it became clear that kidney disease was the prerequisite for NSF. And this common denominator has held true since our first publication, and was critical for focusing the search on substances that could accumulate in the body due to renal disease. I've written about other theorized comorbidities, clotting tendencies, vascular surgery, erythropoietin use, and others. And other authors have proposed acidosis or pro-inflammatory states as explanations. In the end, the simplest explanation that can explain every case of NSF is the best. The current working theory of gadolinium administration in the setting of kidney disease is simple and can be identified in virtually every case of NSF. What's more, the contraindication of high-risk agents in the setting of kidney disease has virtually eliminated new cases of NSF. There is hardly any better evidence than that. Can we say we have eliminated all cases of NSF? Not with certainty. Can we say we have so reduced the number of new cases of NSF that they are no longer detectable in the population? Probably. Question 19, quote, I truncated this a bit because it was quite a long question, but it starts in as, since human in vivo comparative studies confirmed that transmethylation occurs in healthy people, wouldn't this fact alone be proof that gadolinium-associated systemic fibrosis could potentially happen in all patients, including those with normal renal function? End quote. This is a tough question because of the word could. Could NSF occur in a patient with normal renal function? Well, as scientists, we try to remain open to that possibility. My colleagues and I recently developed a set of clinical and pathological features of NSF that could be used to diagnose the disease while eliminating disorders that simulate NSF. In that definition, we chose not to exclude patients without renal disease from being diagnosed with NSF. Stated another way, even though every case of NSF I have seen to date has some degree of kidney disease, we did not insist that kidney disease be a prerequisite for making the diagnosis. We would very much like to see a case of true NSF without kidney disease. Why? Because that case may carry a clue that helps to better understand what is going on in NSF. Could NSF potentially happen in a patient with normal renal function? Based on the millions of doses of gadolinium contrast agents given to patients over the years who have not developed a disease, my answer would have to be not very likely. Question 20, quote, also a truncated question. What research is being done to understand possible links to systemic fibrosis or other harm that can be caused from the longer-term exposure to gadolinium in patients with documented levels of gadolinium toxicity due to multiple contrast MRIs over a prolonged or compacted time frame, end quote. This is a bit confusing. I think the questioner is asking what research is being done to understand the risk of long-term gadolinium exposure due to multiple contrast administrations. I will simplify this a bit more to simply ask what research is being done to understand the risk of long-term gadolinium exposure. As I mentioned before, there is no central clearinghouse of research projects. Unless someone shares with me that they're tackling a particular question, I have no way of knowing what study is underway until the work is actually presented at a conference or in a paper. I can say that there are many entities interested in minimizing long-term risk of gadolinium exposure, not only because it is the right thing to do, but also because they do not want to be held financially or criminally responsible for neglecting to do so. I do not know what techniques they are using to monitor the long-term effects other than those mandated by the US FDA or the European Medicine Agency. As mentioned before, purposeful administration of gadolinium to patients at risk for NSF is specifically contraindicated. Question 21. Quote, there has been some success with treatments such as third-generation Gleevec, electrophoresis, and chelation. Could you please explain how these treatments work? End quote. There have been numerous small studies and case reports purporting to show positive effects from a variety of therapies. I think it is safe to say that none of these works in all NSF patients all of the time. If they did, we would not need to have this session. The study of treatment responses has been challenging for many reasons. Here are a few of the more common issues. 1, the number of patients in any given study ranges from 1 to a few at most. It is very difficult to draw meaningful conclusions from studies of this size. Number 2, the studies are largely conducted without control groups, so the effects of treatment cannot be distinguished from the effects of chance. Number 3, expensive treatments, such as extracorporeal photopheresis or ECP, have not been funded at a high enough level to ensure a full course of therapy for study participants. 2 therapies can be recommended without reservation on my part. 1, physical therapy with the goal of preventing contractures and maintaining flexibility, and 2, renal transplantation for those who are candidates for this procedure. Any medication has the potential to stir up new problems. Had radiologists only embraced this concept earlier, we might not be having this discussion at all. The pros and cons of all potential treatments need to be weighed carefully before proceeding. The questioner does mention 3 treatments for which there has allegedly been success: Gleevec, photopheresis, and chelation. For the first 2, I am aware of reasonably well-conducted small studies that suggest there are benefits to treatment in some patients. I'm not aware of chelation as having been successful in the treatment of NSF. The details of how these agents might affect a change in NSF is beyond the scope of this briefing. I will refer the interested reader to pertinent publications to obtain this information. Question 22. Quote, in addition to having fibrotic changes, many GASF patients at some point exhibit classic symptoms of mastocytosis, severe burning, itching, skin splotches, and dermatographia. Is a possible link between gadolinium exposure and mast cell activation currently being explored? End quote. This is a very astute observation. Several people have asked me about it over the years. In the original investigation into NSF, before the first publications, my co-investigators and I specifically examined biopsies for the number of tissue mast cells. These were not noted to be present in larger numbers than normal and did not seem to look abnormal in any way. This does not exclude a potential role for mast cells, however, and I believe a well-constructed study might answer some outstanding questions. To my knowledge, nobody is currently pursuing this line of research. That concludes the responses to the questions submitted to the Global Fibrosis Foundation webpage. The opinions expressed in this session represent the informed interpretation of a body of work relating to NSF published over the last 12 years. In the spirit of investigation and discovery, these interpretations may be subject to revision if new, scientifically sound information becomes available. A written transcript of this session will be placed on the GFF website for future reference, and the video recorded during this session will become available in coming weeks. Please visit our website for additional information and references. If you support the work of our organization, please consider a tax-deductible donation. Thank you.